Okay, welcome everybody, and thank you for joining us today. It looks like we have a pretty good group in here, and if others join in, they'll be able to join in as we go along. So let's get started this morning with the OsmoTECH HT demonstration, and we'll get moving along here. First, a little bit of housekeeping to take care of. We'd love to hear your questions, so feel free to enter them in the chat at the top of the Teams menu there, as you can see the chat bar. And send us your questions anytime throughout the presentation or the demonstration. We'd be happy to get to those questions either during the presentation, or we're going to have time at the end of the presentation as well in the demonstration to go through questions. So before we get started, we have a quick blinded poll question we'd like you to answer for us. So please see it pop up on your screen, answer that question for us, and then we can get started with the presentation. Looks like we have some good answers coming in. Thank you, everybody. And now we'll move on to the next portion of our presentation. I think I was... Okay, I had a little gap there. All right, to begin with, we'd just like to talk a little bit about Advanced Instruments. I'll give you a brief introduction to the company. The company is a 65-year-old leader in osmolality testing. We also are experts in cell line development technology and liquid handling quality assurance. Advanced Instruments developed the first freezing point depression technology instruments all the way back over 65 years ago. And to be just an interesting note about that, it was actually designed to test milk initially. It was freezing point detection of milk to check if farmers were adding water to their milk. We then developed that technology for osmolality testing in the clinical market space, and we have been developing and advancing the technology to meet the needs of the biotech world ever since that time. We have customers in over 90 countries throughout North America, EMEA, and Asia-Pac region. We are headquartered in Norwood, Massachusetts, with offices in Westbrook, Maine, and Winborne in the UK. We have direct offices in EMEA, in Germany, Switzerland, France, Austria, the UK, and the Benelux. Outside of those particular countries, we are supported by our distribution network, and we have distributors in over 90 other countries in the world. So, Advanced Instruments currently offers 4 different product lines that are utilized in the biopharmaceutical space. Today, we're going to talk about the OsmoTECH osmometers. OsmoTECH is the only osmometer specifically designed for the biotech market and optimized for testing the more complex biological samples that we see today in the marketplace. We also have our Solentum product line, which is our cell line development, which is used for single cell seeding and proof of clonality. We have our Inoxamat product line, which is used for culturing anaerobic bacteria in jars, allowing to isolate your anaerobic bacteria. Finally, we have our Artel product line, which is used for calibrating of pipettes and automated liquid handlers and doing volume verification to assure that your assays have the proper volume to assure good outcomes. Okay. Now we're going to focus on the OsmoTECH osmometers. Let's discuss some of the applications of osmolality in the biological development processing. Starting with cell line development and through cell culture scale-up, osmolality can be used to confirm concentration of media, feeds, and help you gain insight into the scale-up steps. In downstream purification workflows, osmolality is used to confirm buffer concentrations and help ensure buffer exchange and measure the concentration of the drug substance. And finally, in formulation, all the way from development through fill and finish, osmolality is a critical quality attribute that provides valuable information about the identity, safety, purity, and stability of the drug products. For injectable drug products, the osmolality of the solution must be in the physiological range, or it must be close to the osmolality of blood, since a higher or lower osmolality can cause pain at the injection site and by creating a hypertonic or hypotonic environment. Well, now that we've introduced the different use cases of osmolality, here's another blinded poll question. So if you could take time to answer this for us, we'd appreciate it. Okay, looks like we have our responses coming in. Thank you very much, everybody, for answering the question for us. And we will now move on. When selecting a new osmometer for your lab, these are some of the features that you may consider, but this is in no particular order. Obviously, we want to make sure the testing process is simple, the instrument is accurate and reliable. How much volume does it need to run a sample? So do you need a small sample volume of, say, 20 microliters or something, or can you have a larger sample volume that may, for your particular sample in your process? How long does the test take to run? It's very important. How do we record the data how do we record the data we generate in the instrument? So all of this will be taken into consideration when we look at the OsmoTECH HT, and we'll show you how these will all fit into your particular workflow and your regulatory compliance issues. So this is the OsmoTECH portfolio of osmometers, and they're built specifically for the biopharmaceutical labs. We took the customer feedback and created these instruments to meet the needs that you have in your regulatory and complex environments. The testing process is fast and simple and easy to integrate into any workflow. They have flexible data management connectivity options, and strong data integrity features. These features are adjustable to your lab's specific needs and are flexible for R&D, process development, manufacturing settings throughout the workflow. We also have the OsmoTECH XT, which has the largest osmolality range in the marketplace of 0 to 4,000 milliosmoles, which is optimized for very viscous and highly concentrated samples. So that's the OsmoTECH line of osmometers. Though, one other note I'd like to make is that all of these osmometers meet all of the pharmacopoeias around the world. So they all comply with the US, European, Japanese, Chinese, and many more pharmacopoeia standards around the world. So today we're going to look at the OsmoTECH HT, which is our automated high throughput osmometer, where you can test up to 1,000 samples without any maintenance or customer interference. We're going to spend time showing you how to run the samples and how well the OsmoTECH HT works with your data integrity and data compliance needs and guidelines. As you can see, it has a 0 to 2,000 milliosmol range. The test time is around 90 seconds. Sample introduction is through a 96 well plate format. You can run up to 1,000 samples before having to do any maintenance to the instrument. 40 microliters of sample is required in this particular unit, and you can put up to 180 in if you're trying to do 3 replicates of any particular sample. This can be used in applications such as upstream cell culture, formulation, or QC, and again, it supports all the pharmacopoeia guidelines such as USB, EP, and Japanese pharmacopoeia. So before we begin the demo, just want to remind everybody that if you have questions, please put it in the chat, and we'll be happy to answer those along the way, or we'll get to them at the end of the full demonstration. So if you hold on, I am going to switch cameras here, so I'm going to need to turn off my camera first. Need to stop sharing. Need to select a different camera. Okay. And I will spotlight this for everybody. So hopefully you're seeing.
Rick, that looks good on our end.
Does it look good? All right, great. Thanks, Jared. All right, let me move around front here. Nope, I'm not in the picture. I'll move this side. My head's cut off. Okay, so this is the OsmoTECH HT. Sorry that my head's, I'll dip down a little bit. The OsmoTECH HT osmometer. You can see here, we have a touchscreen display here where you'll input all your information for your testing. You have the belt here where your plate will go on. So we have our 96 well plates, which will go in there. We have the hood over here on this side, which is where all your osmometer module is at, and your cleaning and your 96 well moves into there for testing. So now I'm going to move back over here. Let's go to a different camera. Hopefully everything will work. There we go. All right. So just another quick overview. You have touchscreen display, our belt. Up here at the top is the hopper. The hopper is where you load your sample cups in. You load 1,000 sample cups at one time, and you never have to load them again. Over here underneath what we call the hood is your cleaning reel. We'll get a better look at this, your pipette, and where your sample cups come in for testing. Down here below, we have our waste container where the used plates go. You have your cleaning solution, and you have your waste bottle down here at the bottom. Again, everything will last 1,000 tests before you need to change it, so you never have to worry about that. Now I want to quickly turn over to this side and talk a little bit about the consumables that go with the instrument. So we have our bag of 1,000 sample cups here. We have our wiper ring, which goes in to clean the sample probe between each test. We have our system cleaning fluid, our hazardous waste bag, which goes in the disposal bin. We have our box here of this is our test kit. So you have 11 96 well plates in here, 22 evaporation covers, 11 probe cleaners, and then one hazardous waste bag. So these are your evaporation covers, which will go on top of the 96 well plate. These are your 96 well plates, which have a barcode on. The barcode is read every time you run the plate. That allows for you to keep reusing the plate, because the instrument will remember what positions have already been used, and then you can just reuse the plate and continue to use it until you have used up all 96 wells. We also have a calibration cassette here, which is used for calibrating the instrument, and also comes with a verification position so that you can run your calibration and then run your verification at the same right after the calibration is run. We'll talk a little bit more about that. You have a calibration kit here as well, which comes with all the calibrators need to calibrate the instrument and the reference solution 290 to run the verification at the end. Okay. So now let's take a look at the little closer up of the instrument here, and we will talk a little bit about how we want to go about running a test. So what we have here is we have the image of our plate here. We have the image of our plate here, and when we go to load the plate, we will select which positions we're going to run in this particular plate, and it runs through a sequence here that'll show us how to do that. I'll show you that in a minute. We also have our counter here, which tells us how many sample cups are left in the hopper. Also the number of cleaning, the number of cleaning, the test cleans that we have on the roll. It shows you how many plates have been loaded into the system, and it shows you the values of how much cleaning solution we have and how much the waste bottle is full. You also have a stat button up here. If you can run stats. I'll show how that's done later. Our menu button, we'll get into the menus in a few minutes here. So I'm going to jump back over to the other side here, and we're going to quickly load a sample into the instrument. So what we're going to do is we're going to take our 290 reference solution. I'm going to put 40 microliters of sample into position A1. Position B1. Then, although we're only running 2 samples, if I was loading more of the plate, I would obviously need a evaporation cover would be much more important. But in this particular case, I'm going to show you how to put the evaporation cover on. The evaporation cover has 2 little tabs at the end. You peel off the middle section. You'll align the evaporation cover over the 96 well plate. press it down. We have a little roller that comes with it just to make sure it gets rolled on tight on there. You peel off the little tabs. And then we're ready to load it onto the instrument. I'm going to, should have brought my remote control. I'm going to go back to position 2 here. So now what we want to do is we want to load the plate onto the instrument. Now you see the barcode that I mentioned before. You want to take the barcode and make sure it's facing in, because there's a barcode reader right back here, which will read the plate, and that will determine that they will read that plate every time we put it in there, and it will know which positions have been used and which positions haven't, so that we can continue to reuse the plate. So to start running the test, what I'm going to do is hit the load button. The plate will move into the instrument, read the barcode there. It's verifying the plate, checking to see if any positions have been used. You can see it says loading here. You can see the plate hasn't been used before. You can see the date and the time at the top here. This instrument has also been named. You have the option to name it whatever you want. So I'm going to select A1 and B1, where I put the samples in. Then I'm going to hit start. So when I hit start, it's going to ask me if I want to dispose the plate after the test. Since I'm only using 2 positions, I'm going to say no. That way we'll bring the plate back out to the loading area so that I can reuse it. It's going to ask me how many replicates I want to run per well. I'm going to only run 1. Continuous plate testing, I'm going to say no, because that would be if I was loading 2 plates at one time. You can load up to 2 plates at one time to run simultaneously, or you can just load 1 at a time. Remote control mode, we are not in remote control mode, so that's fine there. So now the system is going to start to run the test. So what it's going to be doing here, and I should have done that before it started. Oh, hold on. Okay. So now, we're looking in here. You can see the pipette over here is drawing the sample out of the well, or out of the 96 well plate. It's going to move over to the load position where there is a sample cup. You see the turntable turning, it picks up a sample cup. The pipette is going to dispense 20 microliters of sample into the sample cup. And then the turntable is going to move over to this position where the testing will begin. So you can see the pipette is now going through the cleaning cycle. So it's cleaning the inside and the outside of the pipette here. The sample cup is moved over into the testing position. The sample probe will come down into the well, and the test will begin to run from there. You can see on the screen now, it shows that position A1 is now in testing. B is grayed out. That will be the next one tested. Now you can see here that where the status bar is moving across. So we're in the cooling portion of the testing, where we're driving the temperature down below the 0 degrees C and its freezing point. Once we get to the set temperature in the instrument, we'll hear the knocking sound, which initializes the crystallization of the sample. And then we will get our sample readout in the display here. I did not add any sample IDs in this particular case, but you can add sample IDs as required for every position in the well. Now I did this all manually. When we get into the menus, I'm going to show you how to put in templates, which will make this a much smoother and more efficient process. So that if you're doing the same testing over and over again, you pull up the same template, drop it into the system, and it knows exactly what to test every time for you. So we're getting close to the end of the test here. We should get our knocking sound, and then we should get our result very shortly here. There we go. There's our knocking sound. The sample now goes up to its true freezing point. We're monitoring that plateau, as we call it, so that we know so that we're waiting for it to stabilize. Once it stabilizes, we read out the osmolality, and then it'll display on the screen. So the sample probe has just cleaned itself between tests. We got a 292 and a 290, and that's how the instrument operates for every test. Did load 2 samples. I'm going to cancel that out now so we can start to get into the menus. Once it's finished cleaning, then we can start into the process of showing you how to set up the instrument and show you how all the data integrity and data management features will work for you in your particular environment and processes. Okay. So now we're going to hit the menu button. It's still finishing off. No, that's right. It's got to bring the plate back out. That's what I said to do. So you see it brought the plate back out to the start position here so that now we can reuse the plate. You can remove that evaporation cover. That's why we give you extra evaporation covers. And you can then reload samples into the plate and start over again with that plate. Boom, back in again. Okay, so we're going to hit the menu button. We're going to start out by talking about some different parts of the system here. So we're going to go into the settings menu first. And what we want to first talk about is adding users to the system. So the system can be set up to have a usernames required and sample IDs required. In this particular case, we're just going to go, oops, hit the wrong button. Users. Enter. Entering my password. So every time I go into menu item, part of the audit trail and part of the data integrity is to assure who is in that menu and who's doing what. So every time I go into a menu, I have to put in my username and my password. This assures that the audit trail is consistent and it shows every change that's been made and by who it was made. There's 2 ways to add users onto the system. The first way is to add the users here. You put in a username, a password, which then they would have to change, and then you select the access level they have. There's 3 levels of access on the instrument. There's an operator, a supervisor, and an administrator. I'm logged in as the administrator right now, so that I have access to all of the functionality of the instrument. If we wanted to save that, we would save it and then send it off to the person to use. We exit out of that. The other option that you have is to use LDAP or LDAP-S. What this allows you to do is to put in, to use your Windows login or whatever login you use every day to log into your work computer as the login on this instrument. Your IT department would come in, set up the access level groups, administrator, supervisor, operators, put in people's names and passwords that they use every day, and then they would use that to log into the instrument. So that's another way. This passwords here, this is the password. This is where you would set the password strength and if it's going to expire. If you're adding the users on the instrument, if you're using LDAP and you're using your Windows login every day, that's still managed by your IT department. It wouldn't be managed here on the system. So we'll exit out of that. And that's how you add users to the system and gives you your different access levels for different users on the system. Next, I'm going to show a little bit about how we set up the instrument for testing. Again, logging in to show that who's logged in and who's making any changes, if we make any changes. In this particular case, this instrument, when I first set it up, you notice I did not have to log in when I ran that test. So right now it's not set up to require user IDs or sample IDs to run a test, which is the most basic way to operate the system. However, if you're looking at a GLP or GMP environment, you're probably going to want usernames and sample IDs required. So I'll show you how to set that up in a minute. So here's the user accounts. You see we have our supervisors and operators. Since I'm logged in as the administrator, I can choose what they have access to on the system. So I could have it such that only thing that the operators could do is run tests, for instance, or I can have, as you can see, supervisors have a lot of access here. I can make changes to this. So let's say I don't want the supervisors to change the language. I hit save. Oh, it's not turned on. Okay, I'll get back to that. So normally what we'd have to do is turn on is reason for change, and I'll show you where that is later in the setup menu, where it would require me to put a reason why I made that change into the system. So I'll show you where to turn that on, and we'll go through and do another test there. So what we have here is requiring user ID and sample ID for testing. I'm going to turn both of those on. I'm going to hit save, so we have that turned on. So this now will, if we go back out and we try to run a test, it's going to require me to log in again as in whatever user I want to log in as, and then it's going to require me to put in a sample ID when I try and run a test. The final section here is auditing. Auditing is electronic signatures on the instrument. We have the capability to have a review and approval. You can do one or the other, however your system works best. You can have somebody review all the results and then that's fine, or you can have review and approval, or you can just have approval of the results. The system is smart. It will not let the person who ran the test approve or review their own results. If they try to go in and sign off on it, it will not bring up their username, and you have to log in to review or approve results, and their username will not come up if they tried to do that. So here's where you require reasons for all changes here on this section here. So I'm going to turn that on and save that. And then I'm going to go back to the user access level. If I can hit the button right. And there we go. And I'm going to make another change just to show you how this works here. Hit save. And you see it came up with the reason for change. I hit enter, and now I put in a reason for change. I always recommend that you turn this functionality on, because if you're looking at the instrument and you ever get audited, the FDA or whichever auditing agency you're working with is always going to want to know why a change was made to the instrument. They're going to look at the audit trail. They're going to say, you changed this on this date. Why was that change made at that point in time? If you have these reasons in there, it makes your life much easier if you're ever in that audit situation. We'll exit out of this particular menu. So that's the restrictions. That's how you set up the testing on the instrument. Makes it very easy to run through your testing, very easy to work with the system. Let's talk about data management now. We talked about data management and being compliant with different regulations and managing your data and how you store your data. So in this particular section, we will look at how you are able to manage your data on the system or through the system. There we go. So this is the export function. You're able to export any of the 4 databases here, the results database, the users, the controls, or the events database, off the system as a CSV or PDF file. The events database is our audit trail, so that can be sent out as well. You can send it to a USB. There's numerous USB ports on the back of the instrument. There's also one on the front up here, or to a network folder. You can require logins for export if you want, but most of the time it's just easy to export the data out. One thing to understand about this is there's unlimited data storage on the instrument. That means for the life of the instrument, you will be able to store all your results, all your event logs, all your users, all your result or controls on the system. It will never fill up the database. So it gives you unlimited data storage so that if you're ever in the audit situation and you the auditor says, I want to see where that test result came from, you can go back to the instrument and show them the original source of data, which is always what they want to see if there's any questions about any particular data points. You have the database backup function. Now, the database backup function backs up all of those 4 databases, but it also backs up the configuration files on the system. So having it back up, you're assuring yourself that if there ever is a catastrophic failure, you'd be able to go to the last backup file, import it back onto the instrument, and then you'd be able to start up from that last backup file. So again, you can send it to a USB or a network folder. There's 2 ways to back up the system: a scheduled backup, which would be like once a day, week, or month. And then there's a synchronous backup. The synchronous backup backs up the file between anywhere from any 5 minutes to 60 minutes. And the difference there is it's one continuous file that gets rewritten every time it backs up. So it's up to your IT people or your data integrity people on how they want to do that. Do they want 12 individual files for a year, or one big file that would cover an entire year or the entire life of the instrument, for instance? So there's the import function, result export function. Enable, what this is to enable a PDF report for each run of tests. So you can have one PDF file per test, or you can have one PDF file per run. So if you were running, say, 48 of the 96 wells, you could have one PDF file for every test result, or you could have one PDF file for each run of the instrument. We don't have this turned on right now, but it is a capability. It's like having an electronic printout of the instrument. Finally, we're going to get to templates. Templates is how you set up the testing on the instrument. And like I said, I manually picked the wells that I was going to test when I ran the first test there for you. With a template, you would already have that laid out. So in this particular case, what you're able to do is download a template from a USB or network folder here. And we'll show you where to set up some of the templates. And there's another way to set up templates as well, other than downloading. But you can set them up as a text file in a folder, and then you can download them into the instrument, and they'll download into the positions where you need. Exit out of there. So next we're going to talk about networking. In networking, we talked about managing the data. Now this is how you connect the system so that you can get that data from the instrument to whatever files or folders you need. There's a couple different ways to connect to the system. The first way is through a local area network. Basically, your IT people would set up an address for the system, set up folders. Your results data would be sent to that folder once it's approved and reviewed, if that's the way you have it. And then you'd have an automatic setup of this, or automatic backup of the results database. There's also OPC-UA. Now, OPC-UA is, I like to put like a machine language. OPC-UA allows you a true bidirectional communication with the instrument. In this particular case, you can start the instrument, you can stop the instrument, you can send templates to it, you can send user IDs to it. There's a lot of functionality here. It also helps if you're trying to work the system with other equipment so that you can coordinate between the pieces of equipment and, say, an automated system. We also have a web server. The web server is just a web address on your network, which allows you to review the results on the instrument. It's just a way to review the results. There's nothing else you can do there. You can't modify, alter, or delete anything, which is an important point. All of these databases are unlimited data storage, as I said. Also, the events database and the results database cannot be deleted and modified, altered, or changed at all. They will always reside on the instrument, and you can't make any changes to them. So another important factor when looking at data integrity regulations and standards. Okay, we talked about users, controls, restrictions, networking. The other ones are pretty self-explanatory for other reasons. Let's take a look back here, and let's go into the templates menu and talk a little bit about how we set up a template. So I showed you the one place where you could import templates into the system, either from a USB or from a network folder. And here's where you would start to create your own template if you wanted to. So if we wanted to add a template, we would hit the add button. It brings up our 96 well plate screen here. And then we can choose. So we can choose. I can't hit the button from this angle. Anyways, you can choose the buttons you want. I'm trying to hit from an angle here, and I'm not seeing the angle correctly, but you choose the wells positions you want. You can tell it whether it's a sample or whether it's a control. You can enter sample IDs here. So if I hit enter sample ID, then I would, I'm not doing it from a sample ID. Then I would, come on. So there, it would enter the sample ID, and then I would be able to start running the test. I'm going to exit out of this and not save it. But that's how you would create your templates on the instrument. Again, it's easy also to import the templates from your computer as well. And you can see there's no templates on this particular system. If you had templates in here, you could edit them, view them, you could delete them, everything you needed to do with the particular templates. Let's exit out of this section. So there's the verification functionality too, and what this allows you to do is verify that the instrument is operating properly. So what we do in this particular case is we would take our calibration cassette, which we have here, we would remove the calibrators, and we would put our ampule with our verification standard into the system, and then we would hit the next button. It would go through, it would draw up 5 replicates of the verification standard, and then it would run the statistics for you to show you that the sample was within range of the operating parameters of the system. I'm not going to run that right now, but that's how you would run a verification if you wanted to run a verification on the system. You can also always put controls into your 96 well plate, of course, and you can also, and then you could just, as I showed you on the templates, you could tell the system it's a control, and then you'd be able to put control data into your results database and your controls database as well. So go back in. So we talked about verification, talked about templates. We went into the settings. Just quickly talk a little bit about calibration. So when we do with the calibration, when we talk about calibration here, we're talking about calibration adjustment. There's sometimes some confusion about calibration. Is calibration verification or calibration adjustment? Calibration verification is running your controls on a daily basis. So you take your 290 reference solution, or if you're running a standard at 500, you take, or at 400, you take a 500 and a 300, and you bracket your samples with that. That's calibration verification. What we're talking about here is actual calibration adjustment of the instrument. What we would do in this particular case is, again, we would take our calibration cassette, we would load it in there, we'd hit the load button, we'd start the calibration process, it would run through, it would run those calibrators, it would adjust the calibration curve once it was complete, and then it would update the instrument. You only need to calibrate these instruments once per year. We do not recommend any more than that. Usually with the annual PM, you would calibrate the instrument, and that's all it needs every year. If you're running your verification every day and everything's good, you don't need to run any further calibrations. Great thing about our calibration process is I can exit out of this now. I do not change anything or lose any data. It does not make any changes to the calibration curve until it says calibration complete on the instrument. So we have not made any changes. The instrument's still running exactly as it was prior to starting that process. So the last thing I want to take a look at here, well, there's 2 more things I want to take a look at. It changed before I got a chance to hit the button, is the results database. And this is where all your results are stored. And again, this is an unlimited database, cannot be deleted, modified, altered, or changed on the instrument. And this shows you all of the results that have been run on the system. You can see we have the 292 that we ran earlier today. Shows you the plate ID it was run on, which position. If we had a sample ID, it would have it in there. If I was required to use user ID, it would show it there. You can then sign the results from here. You can print the results. So if you had the review and approval on, you would sign the results from the screen here. You can sign multiple results at one time, so it makes it quicker and easier and more efficient. You can run statistics on results. So if I wanted to choose these top 3 results here, I could hit statistics, and it'll give me the mean, standard deviation, and CV of those particular 3 results. It shows you an export function, and you can exit out of that. You clear those by hitting the little button here at the top, so it shows you the total tests here are 47. You can search the tests by any of these categories here at the top, which looks like an Excel spreadsheet. You can also sort by these. So if you want to sort by user ID or sample ID or plate ID, you could do that. You can export from here, so you could select what results you want to export from here, or you could export the entire database from here. So a lot of options from this particular screen. We'll exit out of that. And finally, we'll just talk about the events database or the audit trail. This is that database. This is where every change to the instrument is logged. And it will show the changes I just made to the instrument here when we were running through the menus. And you can see I was logged in as the administrator. You can see it says change supervisor access to perform cleaning from on to off. And then it shows the reason for change. The other ones, it shows the change, but since I didn't have the reason for change turned on, it didn't show any reason for change. Again, I do recommend having that always turned on. It's a very good feature in case there's any type of audit issues. Same type of functionality you here see, kind of looks like an Excel spreadsheet. You can sort by any one of the columns. You can search by anything in here. Gives you the opportunity to look at anything in the database if you need to search and find it. We'll exit out of that. Consumables diagnostics templates. I think we covered everything we want to cover there. We'll go back to the main screen. And I think that pretty much wraps up our demonstration for this morning. So we have some time here for some questions, if there are any questions.
Hey, Rick, looks like we only have 1 question so far.
Oh, okay.
So quick question. In the manual, the limit amount to measure a triplicate states 150 microliters. In the presentation, you stated 180. Which value should be used in their processes?
So we say, I'm going to have to look at the manual because I thought we always said 180. So it's 180 because 150. So what happens is we're pipetting 20 microliters of sample into each sample cup when we run a test. However, we require 30 microliters of sample to pipette 20 microliters of sample. That's just the way the automated pipetters work. You're always going to have a dead volume, an extra volume in the sample, and then you have to assure that you have enough sample. So we want to have 180 microliters in there so that we're assured we have plenty of sample to run those 3 tests out of the well. So I would say it's 180 microliters is required. I will check into the manual and find out why the manual is different because I know we've been using 180 all along. Okay, any other questions?
Nope, it doesn't look like any other questions.
No other questions. Oh, quiet group today. All right. Well, if there's no further questions, I will say thank you and give you a few minutes of your day back today. And if there's any further questions, please reach out to anybody, myself or your local rep or your local distributor, and they'd be happy to get the answers for you. So everybody have a great day, and thank you very much for joining us today.